Good afternoon, everybody. Thanks for giving me the opportunity to present some of my work for you this afternoon. My work has some relevance to the thesis defense this morning. It works in a similar area, looking at the evolution of. It touches on the evolution of nodulation in legumes. In particular, we're looking at the a particular innovation called the Symbiosome and how it has enhanced the, or we speculated it has enhanced the evolutionary stability of nitrogen fixation in legumes. Okay, let's continue. I think does this work? Oh yes, that works. Okay, so legumes and nodulation. Legumes are one of the world's most successful plant families, about 20,000 species, and they can be found in all habitats except the open sea. Most species engaged in a nitrogen-fixing symbiotic partnership with rhizobia. Something that's not widely known, actually, is that most are actually trees and live in the tropics. So you're all familiar with crop legumes, soybean, peas, clovers, etc. And of course, these are important, but they're perhaps unrepresentative of what legumes, most legumes actually are. Okay, the other thing about legumes is that they've been the subject of intense scrutiny in terms of their taxonomy and phylogeny. The genomic age has caused an explosion in this kind of work, and there have been several recent revisions of their taxonomy, and the two I've mentioned here are already outdated because there's new ones. There's actually new ones this year. But what we've been doing with these taxonomies and phylogenies, as we heard earlier from Sally, actually, is we can use them to elucidate critical nodes for nodulation, losses, or gains. So this phylogeny, this figure includes a phylogeny of legumes that has allowed us to reclassify the old subfamily groupings. So in the old days, previous to this, the legumes were three subfamilies: the Papilionoideae, the Caesarpinioideae, and the mimosoids. And what we have now is there's been it was always known that this would have to happen, actually. So this the legume phylogeny working group decided to formalize something that was generally known, which is that this polyphyletic group in the Caesarpinioideae would have to become 5 separate subfamilies. So that would be a smaller version of the Caesarpinioideae and 4 new subfamilies based upon previous groupings. It also necessitated, unfortunately, that the old mimosoid subfamily would have to be subsumed into a recircumscribed Caesalpinioides as a clade. So it's been downgraded to the mimosoid clade. The Papilionoides are unchanged. So the Papilionoides are essentially as they were. What's this? Okay, so nodulation. What do we know in terms of these new subfamilies? So the Caesalpinioides, outside the Caesalpinioides and outside the Papilionoides, we don't have any nodulating genera. So those four subfamilies, Ditarioides, Cercidoides, Duparketioides, and Dialioides, as far as we know, are non-nodulated. We have to be open-minded. There maybe we will find some nodules sometime, but at present, it's generally considered to be that they're non-nodulated. So nodulation then is confined to the Papilionoides, where most genera nodulate, and that's where the majority of nodulation genera are housed. Outside the Papilionoides, in the Caesalpinioides sensu stricto, or at least the non-mimosoid component of the Caesalpinioides, we only have 8 genera as confirmed nodulation. Actually, there's probably 9 now. It's been there's another one recently being described. However, interestingly, almost all the mimosoids can nodulate. So the mimosoids have arisen from a part of the Caesalpinioides and have evolved from them, and yet they're predominantly nodulated. So that's been a mystery as to why that situation has occurred. So just to reiterate that there are about out of the 20,000 legume species, there are about 6,000 that don't nodulate at all. Okay, as far as we know. So that's quite a significant chunk. But so really what we're saying then is that nodulation has occurred at these 2 nodes. So we've got the nodulation has arisen where the Papilionoides and Caesalpinioides have separated. So these are what we would call hotspots for looking at nodulation, evolution. And then there's this one here that I've ringed, and this is the one I'm going to look at in today's talk. This one where nodulation has either never arisen or has hardly arisen or has been generally lost, whereas the mimosoids that come out of them have retained nodulation. So we're trying to answer that question. So quick summary. So 4 basal subfamilies do not nodulate. Nodulation is standard in the Papilionoides. There's an exception in the base of the Papilionoides where it's relatively rare. But nodulation is very rare in the Caesalpinioides, as 9 or 10 genera, depending on which way you look at it, but is common in the mimosoid clade, which is derived from them. Since that phylogeny, there has been a new phylogeny that doesn't actually help us, but I think I should mention it because I say it doesn't help me in what I'm trying to do, but it does give us an idea that, or it makes the idea that there was basal groupings less tenable. So we say that the 4 basal subfamilies, the Cercidoides, Ditarioides, etc., and that the Caesals and the Papilionoides emerge from them. In fact, that's not the case. What we now see is a probable single origin of the legumes, or at least a single radiation of the legumes 60 million years ago, after the impact of the asteroid that killed the dinosaurs, the KT boundary. So the implications of that new phylogeny is that legumes expanded enormously after the Cretaceous-Paleogene boundary, mass extinction. This was accompanied and followed by several whole genome duplication events, and all 6 subfamilies, as far as we can see, appeared simultaneously. So it's hard to define the roots of the origin within the nitrogen-fixing clade. So it could be at the base. There's been a lot of extinctions, for example. For sure there will have been, but it does mean that no extant subfamily should be considered as basal. So basically, they've all arisen at once, or at least what we can see. 4 of them are not nodulated at all. 1 is slightly nodulated with a massively nodulating clade within it, and the other is massively nodulated. So that's an interesting pattern of nodulation that we try to see if we can explain. Sally showed this figure from Griezmann. It reset it restarted a debate that had been started many years ago, actually, but it was generally considered that there'd been multiple origins of nodulation across the nitrogen-fixing clade. And then this paper was published suggesting that there would be multiple losses of nodulation in the legumes and other nodulated groups. And in other words, that there was a single origin followed by massive losses. And particularly these 2 genes were considered to be of great importance. That was NIN and RPG, as it seemed to be. NIN for sure is present in all the nodulating, the ones that are in blue, and tends to be absent in the non-nodulators, although as Sally also pointed out, there are there's a considerable number of exceptions to that. So it's not completely clear cut. However, this hypothesis is the one that's begun to build traction and is considered to be the one that's currently most likely. However, there is a fight back from the other side. So and there's a paper now online which is arguing strongly against this. So it's not cut and dried. And as we discussed earlier, I'm somewhat agnostic. I don't feel I have the capability to completely make a decision either way, to be honest. So I sit back and I wait. However, if we take the view that there's been losses, or we could equally take the view that there's not been acquisition, is just another way of saying it. So how do we explain this patchy distribution of nodulation in different groups within the nitrogen-fixing clade and within the legumes? I think the nitrogen-fixing clade is a big thing for me to try and get my head around. So what we did was we took a smaller grouping to see if we could understand why there have been losses of nodulation in the Caesalpinioideae. This was our first attempt. We just thought, right, let's try for this one. Let's not build Rome in a day. Let's go for the Caesalpinioideae. See what we know about them. In other words, if we take the view that what we see now is the result of losses, the question we would ask then is, why have there not been losses? So why is nodulation stable in some groups, but not stable in others? So in the Caesalpinioideae, nodulation is stable in the mimosoids, in the mimosoid clade, but not in the Caesalpinioideae that from which they arose. So what are the functional factors underlying the evolutionary stability of nodulation? So we used nodule anatomy and whole plant genomes to look at this. That was the initial approach. And then we tried to match nodule anatomy and certain features of nodules onto a new phylogeny based upon 900 core genes. Right, sorry. Nodule anatomy and whole plant genomes can help answer these questions. Okay, so this is a tree from my good friend Domingos Cardoso in Brazil that he put together for me. It's not completely up to date because these phylogenies date very quickly. But basically, the Caesopinioides, you've got the Caesopinioides with the mimosoids arising from it. The blue genera are nodulated, yellow is uncertain, and black is non-nodulated. So it just illustrates what I've said, that most of the Caesopinioides are black and most of the mimosoids are blue, with a significant number of oranges because there's still uncertainty. This phylogeny will be massively out of date even now because they've renamed several of these genera. There's been a massive renaming across the mimosoids. But the basic message is the same. Massive nodulation in the mimosoids and very little nodulation in the Caesopinioides. Now, we wanted to understand why that happened. So we went to what's called the Caesopinioid mimosoid junction, which we consider to be a potential hotspot for evolution nodulation. And that's bordered in the mimosoids with Pentaclethra and Chidlovia, which are both nodulated. And on the mimosoid side and on the Caesopinioid side, this African tree called Erythrofleum. So we focused our efforts on these. I've highlighted two here. That's Mimosa, which is probably the most familiar mimosoid. And I'm not sure why I've noted Adenanthera. Adenanthera is a commonly known non-nodulating genus. Okay, so what do we do with anatomy? For many years, I've been looking at nodule anatomy, particularly with regard to a phenomenon called the fixation thread, which is regarded as a primitive feature. So most nodules have their bacteroids surrounded by a symbiosome, which is a membrane. So if you're familiar with nodules on pea or soybean, the bacteroids are surrounded by symbiosomes. But in the case of these plants that we're looking at here, the bacteroids are surrounded by cell wall material, and they're called, they resemble an infection thread, but they're not an infection thread. They're something related to that, but so we call them fixation threads. It means that the bacteroids within them are essentially apoplastic. They are well described in Parasponia, which is the only non-legume which nodulates with rhizobium, and has been reported for more than 30 years as being the dominant type in the Caesopinioides outside the mimosoids. There's an interesting exception that's in a genus called Chamicrysta, which I won't expand upon that because it's another little story in itself. They are present in a few of the Papilionoides, but their position taxonomically is a bit strange, more towards the base, but not completely at the base. Again, I'm not going to talk too much about them. Crucially, they're not present in the mimosoids. So the mimosoids have arisen from the Caesopinioids, but they are mainly nodulated, but they are not nodulated with the fixation thread type. Okay, so what are fixation threads? You're not familiar with them. Cell wall bound symbiosomes and have been considered as markers of of a primitive nitrogen fixing association. So what they are, so these are bacteria, they're bacteroids, and you can label the wall of the fixation thread with antibodies against pectin. So this is pectin. Another example, these are various trees, legume trees, Chamicrysta, Templetonia, Campsiandra, and Jacksuberia. And they all have the same kind of phenomenon, this wall which surrounds the bacteroids. So these are fixation threads. If we look at mimosoids, there's a couple of examples. This is the case of Vachellia, which used to be acacia, and they have the symbiosome membrane, or at least there's no obvious cell wall here in mimosa, for example. So it's a symbiosome membrane in all the mimosoids that we've examined. So the inference then is there's no fixation threads, always symbiosomes, so they're more advanced. That's the theory. That's the hypothesis. Okay, so our target genera, at least for a detailed study of this phenomenon, was Erythrofleum. It's a tree native to West Africa, and it's the immediate sister to the mimosoids. It has classical fixation threads. So as you can see here, they're surrounded by the pectin containing cell wall. This is the host cell wall. So it's the same material. And if you look at them under more detailed electron microscopy, this is the cell. This is the bacteroid. This is the cell wall. And surmounting the cell wall is a symbiosome membrane. So they do have a symbiosome membrane. And it's derived, we think, from endoplasmic reticulum within the within the cytoplasm. So there is a symbiosome membrane, but it surrounds the cell wall. So we're able to at least determine in more detail that structure. But the main difference is, of course, the cell wall. And the other two genera that we looked at on the mimosoid side was another West African plant called Chidlovia, which nodulates profusely. This one contains symbiosomes. It's really difficult plant to work with. They're just woody things. So there's a symbiosome membrane, and the only time we see pectin is surrounding an invasive infection thread, because we know that the infection thread is cell wall bound, it's surrounded by cell wall, and in Chidlovia nodules, this is the only place where we see a cell wall. The bacteroids themselves are within symbiosomes. Okay, this work was underpinned by a lot of field work, because what we wanted to do, we wanted to use these ones as the examples, but at the same time, we needed to plot the occurrence of symbiosomes, the occurrence of fixation threads across the new phylogeny that we generated of the Caesarpinioideae. And there was a number of things that were missing that were quite key. So it necessitated field work. So this was aimed at filling gaps in our knowledge about nodulation in tree legumes in the subfamily Caesarpinioideae, focused on centers of radiation in the Brazilian Amazon and West Africa. This is where a lot of these trees are very diverse, and that's the place where you've got to go and search for them. Okay, so our first trip was to Manaus in the center of the Amazon. So we sampled plant DNA, so we can identify the plants, and nodules for anatomical analysis and rhizobial isolation. I'll just give you a bit of flavor of fieldwork in Brazil. It's quite challenging, but it's fun as well, but it is challenging. So basically you go up the river in a boat, and as you can see, it's the Amazon has a number of communities living alongside it. So you stay in those communities, and home is a hammock and a house on stilts, and food is piranha, which we try to eat them before they eat us. And then the actual sampling, you've got to head up river into the flooded forest. You can't sample obviously from the flooded part, so you find the areas that are still above water, and that's where we start the sampling. And sampling it's just a question of it's a lot of hard work digging. We also need plant samples for identification, so it's above ground and below ground. But in most cases we obtain nodules. This is Schwarzia, which is a basal papilionoide, for example. And one that we were very, very happy to find was nodules on this plant called Jacksuberia, which is in the Caesarpinioide. So that's why we're able to increase the number of nodulated Caesarpinioide. But you get, it's tough work getting it. You get very wet and very dirty. Okay. The other one, another one we discovered there was Pentaclethra. Pentaclethra is known to nodulate, but we didn't know the anatomy of it, and it's the one that's closest to Erythrofleum in the tree in the phylogeny, and it's the nearest mimosoid. So you've got Pentaclethra and Chidlovia. So we needed to know what Pentaclethra, what the nodule anatomy of Pentaclethra was. And pleasingly, we found it was nodulated with the Symbiosome type. So these are the nitrogen fixing cells. This is a nucleus, just for those of you not familiar. These are bacteria. These other cells are we call interstitial cells. These are not colonized by bacteria. Okay, so what we did here, these are also not easy to section. They're big, just very woody. This is a host cell wall. We've immunolabeled it with antibody, monoclonal antibodies against pectin, so it's well labeled. But the bacteroids here are surrounded by a membrane rather than by a wall. So there's no immunolabeling here of pectin. So there's no cell wall around here. So it's a symbiosome as we predicted. But of course, our predictions aren't always we can't be sure they're always going to be correct. But in this case, they were. Okay, so Ghana holds a number of things that we also wanted to check out. There's been some papers published about various Ghana, about West African legumes being nodulated, but they didn't provide much evidence, or they certainly didn't show anything about nodule anatomy. So several of these are either unreported in terms of nodulation, or are considered to be non-nodulated, but with little support. And the key genera were various genera at the base of the mimosoids, Obrivilia, for example, being a good one we wanted to check. There's another species of Pentaclethra, which is considered to be non-nodulated, Piptodeniastrum, and a few others. So these are our target genera in Ghana. So we did a lot of the work in forest reserves. This is the team I was working with. Obrivilia kirstingi was shown to be nodulated. It's previously been reported as not nodulated, so that was very pleasing to find that we got nodules. Piptodeniastrum, previously reported as nodulated, but we don't know about the nodule anatomy, so we got good nodules. So major findings from these expeditions. Confirmation nodulation by Jack Suberia, making it only the 9th genus in the Symbiosome shown to be nodulated. I need to update that because there's actually 10. Dinitia has also been shown to be nodulated, but that's another story. Several collections of nodules from other Symbiosome that we can then check the nodule anatomy. Sampling of nodules from Pentaclethra at the base of the mimosoid clade, sampling of nodules from Obrivilia, Kirstingi, and Piptodaniastrum, and the anatomy of several nodules examined, and the rhizobia to be isolated and genome sequenced. So that's another story, is the rhizobia. We'll tell that story today. So to do the analysis and to track the presence, we needed to presence of fixation threads or some symbiosomes across the Symbiosome. We had to catalog all the anatomical knowledge that's in the literature, or it's in my database, or in the database of my colleagues and collaborators, and which we also obtained from these new samplings. So we examined them anatomically for the presence of fixation threads, FTs, or symbiosomes, with the key genera being Erythrofleum, a non-mimosoid, Pentaclethra, a mimosoid, and data from another 128 species from 56 genera in the Symbiosome. So we plotted these data onto a new robust phylogeny of the Symbiosome constructed using 997 genes. Nodes were dated and losses of nodulation indicated. Okay, so this is a quick, more detailed version of what I'm going to show in a minute, just to remind you what we're looking at. So these are mimosoids, and these are the Symbiosome. Erythrofleum is here, Pentaclethra is here. Okay, Chidlovia is there, marked in green. And Pentaclethra, there you go. Okay, so this is the final result. So this is a phylogeny of the whole of the Symbiosome, as much, almost all the genera are included in this. We've just published it, so I'm happy to share this paper with you, of course, if you're interested. So you're not going to see the details here. But the mimosoid clade, that's the mimosoid clade, which you'll notice is also considerably more diverse than the Symbiosome. So when the mimosoid clade has arisen, it's also expanded enormously compared to its, if you like, its parent lineage. And we don't know if that's linked with nodulation either, actually. That's another interesting possibility. Okay, so what we got then is the Symbiosome type is in blue. The fixation thread type is in red. Green is unknown. And there's a purple one, which is unknown. We know it's nodulated, but we don't know what type it is. Sorry, green is non-nodulated. So I'm talking rubbish. The black is unknown. We don't know what the status is of those. So firstly, you can see that green is present in the mimosoids, but not really very commonly. So we've got some things like Parkia down here and Zapoteca and a few others at the base of the mimosoids. We are considered to be non-nodulated. But if you go into the Symbiosome, most are green. If we know what they are, they're green. And the red ones are, well, there should be 8 or 9 of these. So that's the other thing we show, is that when there's nodulation in the mimosoids, it's a blue type, it's a symbiosome. When there's nodulation in the Symbiosome outside the mimosoids, its fixation thread is red, with this weird exception in Pentaclethra. So we wondered if the fixation thread was associated with the nodulation losses. So it seemed to be, if you house your bacteroids in a fixation thread, do you eventually lose nodulation? And we were able to look at the transitions, and we showed that nodulation was indeed lost. Was the prevalence of losses of nodulation was statistically greater in the, so this is just to highlight this, was statistically greater in the Symbiosome outside the mimosoids. So if you have a fixation thread type nodule, you're generally also associated with nodulation losses. So the green type is also is often sister to the red type. Whereas if you go up into the mimosoids, apart from this group at the base, nodulation is generally stable, and it appears to be associated with the symbiosome. Not shown here is we also showed that nodulation rates of losses of nodulation, not just incidence, but rates were also higher in the Symbiosome. Sorry, that's the. So we just published this work in New Phytologist, and we have the final published version available just last week. So conclusions. So using nodule anatomy and a robust legume phylogeny, we have demonstrated that the adoption of symbiosomes by mimosoids could be seen as a key innovation, allowing for stability and persistence of the nodulation traits in legumes. We think that this is because symbiosomes provide intimacy and compartmentalization. In other words, they represent a major investment by the plants. So the fixation thread is kind of the plant, what we call expression in English, hedging its bets. Do we want you or do we not want you? You know, and if they don't want them, they can remove them quickly. But the symbiosome is something, it's symplastic, it's internalized within the cell. There's no barrier between, almost no barrier between the bacteria and the plant. So the plant has completely invested in this almost organelle type relationship. Pierre-Marc Delot and his group have just sequenced the genome of Varififlaum, which we're hoping is going to give us a better understanding of this relationship. Not just in terms of losses of nodulation, but how they might be linked with the evolution of symbiosomes. And we now have the data to also examine the papilionoids, which is going to be quite a challenge. Okay, I'd just like to acknowledge my funders. That's the Bill and Melinda Gates Foundation through the ENSA project at University of Cambridge. I've got a lot of collaborators. The names of the authors of that paper, but special mention to Sergio De Faria, who's been working on this for decades and whose databases of nodulation are used extensively. And I'd like to thank you for your attention.